This simple protocol allows one to label EVs and monitor them via confocal microscopy. For instance, EV imaging might help in evaluating EV distribution when used in therapeutics. This technique allows one to monitor EV migration and uptake in cartilage explants easily, with confocal microscopy without any special function. The same protocol can be used for EVs for in vitro or in vivo experiments. Make sure that the columns are prepared beforehand and that the initial volume of EVs is enough to reach the desired final particle concentration, considering that the around of 10% of the particle will be lost during the purification step.
Begin by concentrating the extracellular vesicle, or EV, sample and the control. Place the samples in a 15 milliliter or 500 microliters concentrating tube, depending on the starting volume of the EV sample. Centrifuge the tubes according to the manufacturer's instructions until an almost dry sample is obtained. Resuspend the concentrated EV samples with 200 microliters and the control group with 100 microliters of diluent C and transfer them to new 1.5 milliliter centrifuge tubes. Separate the EV sample into two aliquots of 100 microliters each. Mark one of the aliquots with dye and use it as treatment. Leave the other unmarked, but process it like the EV sample and use it to quantify the EV concentration by nano.